Everyone. Good afternoon. And again, I'm Dr. Williams introducing our speaker. It's really a privilege to listen to metabolic mechanisms of myocardial hyperemia and implications for exercise and the aging heart. We have Dr. Professor Bradford Hill here from University of Louisville. He is a university scholar, and he got there by going to college at Rebecca University in Nashville, followed by a master's degree in biology at Fisk University in Nashville. He obtained his Ph.D. in biochemistry actually here as a Louisville Cardinal, and he was a postdoc student and then joined our faculty as an assistant professor in cardiovascular medicine in 2009. He actually has worked his way up in a timely fashion through the ranks to full professor and tenured, as well as being the university scholar, as I mentioned. He has so many publications and so many presentations and a lot of grant funding. He is truly the exemplary university scholar, and we're really thrilled to have you here and thrilled to listen to what you have to teach us today. Research Grand Rounds. Thanks for coming. All right.
Thank you, Dr. Williams, for that very nice introduction. Today, I wanted to try to keep this somewhat short, so I only have about 35 slides or so, but it maybe gives us some time for discussion at the end. And what I wanted to talk about was some of the newer work that we're doing and doing in the center, as well as this is what I'm going to show today is really the work of many investigators that have come together to really try to understand that mechanism of myocardial hyperemia. So as for me, these are my major domains of interest. As you notice, each one of these bubbles has metabolic at the beginning. So I'm a metabolism fiend. That's what I really enjoy studying. We have studies ongoing to understand metabolic mechanisms of heart failure and with respect to fibrosis, and trying to understand how metabolism within fibroblasts may regulate the fibrotic response after MI. We've had papers and some grant funding trying to understand how metabolism controls exercise-induced cardiac remodeling, which today I'll kind of, some of the work alludes to this as well. I'll show, and then more recently we got our first aging grant from NIA to really understand how metabolism regulates myocardial blood flow in the heart and how this is affected by the aging process. And really, this is a group effort. The grant that really funds this work is a MPI grant between Dr. Matt Nystoriak, who was at the university. He recently left to go to Masonic Medical Research Institute in New York, where we're still really actively working. And we joke that we actually talk more now than we did when he was just one floor below me. And also this work has led to some collaborations with Dr. Cholera in cardiology, who's doing some clinical studies that's related to this work as well. So this graph that I have at the left is really the paradigm that we're trying to understand. It's well known that the heart, that blood flow in the heart will increase as myocardial work increases, as demand increases. And this is consistent through all mammals. This particular graph is pigs on a treadmill, where myocardial oxygen consumption was measured and correlated with correlated blood flow. But this kind of relationship has been known for a very long time. You can make a stretch saying it goes back into the 1700s by the Scottish surgeon John Hunter, who said that blood goes where it is needed in his treatise on the blood, inflammation, and gunshot wounds, published in 1794. Of course, they didn't know much about it. They knew that blood seemed to go where it needed. That's about the extent of it. But since then, really what a lot of work has been done is truly trying to understand what is the mechanism behind this relationship. And the best going hypothesis is called the local metabolic hypothesis, where it's thought that changes in cardiomyocyte metabolism leads to the production of metabolites that can promote metabolic vasodilation of arterioles. And if anybody wants to understand coronary flow in the heart and all the basics of it, this review that I cite at the bottom here is just really a true tour de force. It's one of those, it's an example of what a really good review should be. In this paper, this is one that I took out of that one sentence I took out, despite over 50 years of extensive research, precisely how coronary vascular resistance is so tightly coupled with myocardial metabolism remains one of the, if not the central question of coronary physiologists to this day. I'm by training a biochemist interested in metabolism, and I've studied a lot of cardiac metabolism since I became faculty. And this is kind of a diagram showing what the heart uses as a substrate. The heart needs enormous amounts of ATP to continue its incessant pumping action. And in the heart is generally fueled by fatty acids. 40 to 70% of the ATP is derived from fat oxidation. 20 to 30% is thought to derive from glucose. And then lactate and ketone bodies can also contribute quite largely to ATP production. In fact, lactate, for example, if you exercise, it can get up into 10 millimolar levels or so. And when it's that high, the heart will predominantly use lactate for energy. And ketone bodies have been shown to be really important and have come to the limelight recently. This is an example of one paper showing that the testing of these ketone esters in patients with heart failure, showing some really positive results. Other substrates that the heart can use, it really doesn't use BCAAs or glutamine that much. It's actually a glutamine producer. Pyruvate, this is commonly in our in vitro studies. We have high levels of pyruvate in the medium. In circulation, actually, though, it's less than 150 micromolar. So it's not a predominant substrate of the heart in vivo. And acetate's only used, acetate goes up a lot when we drink alcohol, converted to acetic acid, and the heart will gobble up acetic acid into those conditions. But otherwise, it's really not an important substrate. So what are ketone bodies? Ketone bodies are these carboxylates that are produced in the liver that derive from fats originally. And through a series of enzymatic steps, it makes acetoacetate shown here. And this acetoacetate is converted to beta-hydroxybutyrate by the BDH1 enzyme in the liver. Beta-hydroxybutyrate is the predominant ketone body in circulation. And after it's made in the liver, it can exit and go into circulation and enter into peripheral tissues that need it, such as the heart. And then it's broken down by the same enzyme, BDH1, as it makes it. And this can be used for energy in the heart. I'm going to refer to this as 3-hydroxybutyrate. It's got several different abbreviations in the literature, which can be confusing, but I'll refer to it as 3-hydroxybutyrate going forward. Let's start with the human data that's out there. This is an example of, it's a table of data from a study in which HFrEF patients and age-matched controls were infused with either saline or 3-hydroxybutyrate. And then by PET imaging, they looked at myocardial blood flow, which is what I'm going to direct your attention to that. And what they saw is that both in the HFrEF patients and the age-matched control, they saw an increase in myocardial blood flow. But you'll notice that a lot of other things changed as well. External work, MVO2, so the heart's using more oxygen, cardiac output increased, stroke volume increased, mean arterial blood pressure typically decreased, heart rate increased, and systemic vascular resistance decreased as well. But this isn't just a standalone study. There's other studies as well in humans where they've done similar types of experiments. This is an example in just normal subjects showing a really prominent increase in myocardial blood flow in patients infused with beta-hydroxybutyrate. And it did, independent of vascular territory, it increased this blood flow. So ketone bodies have really come to the limelight recently. They have thought to have a lot of beneficial effects. They've been shown to decrease inflammation, to have effects on oxidative stress, to decrease systemic vascular resistance, modulate the immune system. They've been thought to have effects on body weight and cardiomyocyte function. They've been thought to decrease fibrosis, but they've also been shown, as I showed, to increase myocardial blood flow, as shown in that previous diagram. But it's unclear whether this actually happens in mice and whether we can use mice as models to study this interesting phenomenon. So for this, two really talented faculty members, Yibing Nong, who's a service track faculty member in medicine, is just an excellent surgeon and a wonderful physiologist, and Matt Nasoriak, who's a vascular physiologist, came together to really standardize this protocol for measuring myocardial blood flow in mice. So in this pressure volume catheter is inserted in the right carotid artery, where you can get hemodynamic data, and then you have an infusion catheter that's through the left jugular vein where you can infuse whatever you want to infuse, but in this case we infuse contrast agent, which is similar to the Divinity dye that's used commonly in clinics to measure myocardial blood flow. And so the benefit of using this type of an agent is that it can be, these bubbles can be burst, so the echogenic dye is released, and then you can lose the signal in the myocardium, and then you can look how fast this contrast agent replenishes into the muscle tissue itself. Over time, you can plot the velocity, how fast it replenishes, and the overall volume of replenishment. And by plugging this into a basic equation, you can calculate myocardial blood flow. This is an example of one experiment in mice in which mice were infused with dobutamine. And compared to baseline, you can see a remarkable difference in not only the velocity of replenishment, but also the total amount of contrast agent that went into the myocardial wall. So we wanted to see does 3-hydroxybutyrate actually increase perfusion in mice. So we gave a bolus dose of 3-hydroxybutyrate and then give a sustained infusion after that to kind of peg it out at around 3 millimolar levels. So that's a really kind of a physiological level of ketone bodies that doesn't get anywhere near ketoacidosis, but it does seem to have effects in this range in other scenarios. And then you can use the echocardiography and the nonlinear contrast mode myocardial blood flow imaging to look at how much blood flow has changed over time. This is an example of one of the data, exemplary data showing that 3-hydroxybutyrate does increase both the slope or the velocity of replenishment and the overall level of the contrast agent into the myocardial wall. And shown here is an experiment in which we took mice and they had baseline values and then infused the 3-hydroxybutyrate. And you can see that in every one of these mice, we see an increase in perfusion in the heart. And if you plot this as a continuous variable against plasma 3-hydroxybutyrate concentration, it seems to be somewhat of a nice correlation. So how does this actually occur? There's a couple competing hypotheses. One is that 3-hydroxybutyrate could directly promote vasodilation. There's some evidence in the literature suggests this might be a possibility. This is taken from a fairly recent paper in which they used coronary septal arteries, pre-contracted them to roughly 60%, and then gave varying levels of 3-hydroxybutyrate to look at the response. And you can see that the vasal relaxation response to EC50 is around 12 millimolar, which is quite high. And in the range that we're looking at, it really didn't make that much of a difference. So it's just from this, we didn't know this is a possibility, but it wouldn't seem to be that important in the context of what we're studying because it's kind of super physiological. The second competing hypothesis would require metabolism of 3-hydroxybutyrate by cardiomyocytes, and it's thought that perhaps a metabolite then indirectly promotes vasal relaxation. So to test the first possibility, Matt and his lab set up a series of experiments in which they used pressurized vessels that were pre-constructed with either myogenic tone or they used agonist-induced tone, a thromboxane agonist. And then they perfused on 3-hydroxybutyrate at different levels through the physiological range. And what you can see is that there's absolutely no change in diameter in these coronary arteries, suggesting this is likely not responsible for the increase in myocardial blood flow that we're seeing. So to try to test the second possibility, we had gotten mice from Dr. Dan Kelly, who's a cardiac metabolism researcher as well, clinician scientist, and he made this conditional BDH1 knockout mice. And we can breed these with alpha MHC mercury mur mice, which would, once the Cre is expressed, this was expressed only in cardiomyocytes when you give tamoxifen, and would excise exons 3 and 4 of the BDH1 allele, leaving a truncated product that's then degraded. And we can see that these mice work quite well. This is Western blots of hearts of mice that have been fed a tamoxifen diet, either their Cre negative litter mates or the Cre positives. And you can see a marked reduction in BDH1. And this is not, we don't see any compensation by other isoforms of BDH, such as BDH2, and downstream enzymes such as SCOT1 are not changed either. So the model works. So then we wanted to see in this model, is there any evidence that the metabolism of 3-hydroxybutyrate influences the vasodilatory response that's caused by 3-hydroxybutyrate. So we infused these mice, the control litter mates or the cardiac-specific BDH1 mice with 3-hydroxybutyrate, and we got around 3 millimolar levels as we wanted to. And then what we found was that in the control litter mates, the Cre negative mice, we saw a nice increase in perfusion with 3-hydroxybutyrate, but this was completely abolished when the BDH1 was deleted from the myocyte, suggesting that cardiomyocyte metabolism through 3-hydroxybutyrate is required for hyperemia in this context. And because we have the pressure volume catheter in there, and we have a lot of other physiological parameters, we can actually calculate work. And you can see that overall, there's really no change at all in the cardiac-specific BDH1 knockout mice, where we see a nice increase in perfusion and this relationship in the Cre-negative litter mates. So this paper, this work was done a lot by Kara Gallons and Yibing Dong, predominantly the first authors of this paper. And we sent this paper off, and we got a very favorable review, but one of the reviewers says it's unclear if 3-hydroxybutyrate will have the same effects, that is increasing perfusion under disease conditions. So we had this in mind. We weren't thinking of doing heart failure right off the bat. We were actually thinking more in the aging context. And so for this paper, we're going to actually just package what we have because it's showing a really important phenomenon. I think it's better packaged as just hitting that main point. But we have done some work in aging, which I'll show now. We know with aging, it's involved with fibrosis of the heart. There's increased inflammation, diastolic dysfunction, mitochondrial dysfunction, and myocyte and capillary dropout that occurs. But it's also been shown there's changes in myocardial perfusion reserve. This is a study showing about by the age of 50, dipyrimidine infusion usually promotes a hyperemic response. Promotes increases in adenosine. But by about the age of 50, you see that this starts to drop off. This myocardial perfusion reserve starts to drop off. So in the human population, it does appear that this hyperemic response is diminished a bit. So we were trying from the aging angle to ask, might changes in metabolism and myocardial perfusion be responsible for cardiac dysfunction associated with aging? So this is kind of the schematic showing kind of comparing mice versus humans, the age frame, the timeline of age. And you can see, basically where I am 47, I'm a middle-aged mouse, if you will. And our first experiments, we were going to look at young versus old mice with the hypothesis that age-induced changes in cardiac metabolism, which have been shown to occur, alter the relation between cardiac work and perfusion, thereby promoting deleterious cardiac modeling, which we think could be targeted to improve cardiac function over the lifespan. So this is an example data of the myocardial blood flow experiments in old mice. These mice are around 2 years old. So they're probably late 70s if you correlate that to human years versus a young 12-week-old mice, which is a young adult mouse, or young adult human, I should say, correlates to a young adult human. And if you look at this with all the replicates, you can really clearly see that both male and female old mice have really decreased myocardial blood flow in general compared to the young controls. And if you plot this against cardiac work, we can see the cardiac work is actually the same in both, but you just see a decrease in perfusion in the old mice. So we started looking at old mice and just looking, well, could this ketone body mechanism be important in this at some level? And we've looked at BDH1 expression, mainly because BDH1 has been shown to be pretty responsive to stress. So in heart failure, after MI or with pressure overload, you see an increase in BDH1 typically. With HFpEF, that is not the case. But in these old mice, what we see is an increase in BDH1 in both the male and female hearts. And we do see, this is one of the results that we don't see every single time, but we think there's a trend toward a decrease in plasma 3-deoxybutyric in old age. And there have been some publications in the literature of our archive paper that showed something similar, showing a decrease in circulating 3-deoxybutyric, at least in male mice. So taking at base value, it appears that the old heart, the aged heart is really primed to use ketone bodies, but maybe it can't, the liver can't produce as much, possibly. So what we did is we took aged female and male mice and gave them again this 3-deoxybutyric bolus followed by a continuous infusion of 3-deoxybutyric to get around 3 millimolar to 4 millimolar levels, and then measured myocardial blood flow. And what we saw was that in the old mice, again, we see a decrease in myocardial blood flow, but in the ones that we perfuse 3-deoxybutyric, we see that this restores blood flow to near the young levels. So that suggests that that maybe 3-deoxybutyric could be an intervention that can improve perfusion in the context of aging. So old mice are known to be not as functional. These are 2-year-old mice, and they weigh a little bit more. They don't move around as much. They have decreased exercise capacity in general, measured by distance and work. They have functional defects in the heart. They have decreased EF, an increase in IVRT, suggesting diastolic dysfunction, and an increase in LV mass in general. And they have a higher fibrotic burden as measured by hydroxyproline. We were really interested in the exercise capacity component of this. And we thought, well, let's see if ketone bodies restore exercise capacity in aged mice. But what we realized that's a good question, but first things first, what about just healthy adult mice? Does it do anything there where we've already got really know that most of our experiments have been done in the young models? So we wanted to start there. And we thought it may be important because there's really four main factors that contribute to exercise capacity. One, the pulmonary diffusing capacity, the ability of the heart to pump blood to organs, cardiac output, oxygen carrying capacity of the blood, and the ability of skeletal muscles to take up nutrients and oxygen and deliver force. And cardiac output, it's known with exercise, it's really rapidly increased. So if one of you were to take off running down the street now, your cardiac output will go up substantially. And anytime the heart has to work harder, it has to have more blood flow to the heart itself. So we want to ask this basic question. So interestingly, ketone body supplements are a booming industry. There's a lot of companies selling different ketone body supplements. They're very expensive. If you tried them, they taste terrible. But they're peddled at almost a lot of athletic events, biking events, marathons, what have you. And perhaps there's some good evidence that they do improve things. This is what taken off a website of Delta G. They claim increased brain health, increased endurance, heart health, weight control. They claim that it improves cognitive performance and they boost recovery. Kind of a panacea, if you will, which I'm not as attuned to think that it's that, but I think it may have some beneficial effects in general. So if you look at the literature, do these ketone body supplements actually increase exercise capacity? It's really mixed. There's some that say that there's no effect of ketone ester supplementation. Several of these that say there's not. There's some in rats that say that it does. If you go through here, you can get really confused and say, well, why is there some people saying that it is improving exercise capacity, some not? But a lot of this has to do with these are different supplements. There's all different types out there. And one of the most recent ones that have been used is ones developed by Kieran Clark, who's actually a cardiac metabolism researcher herself. And what she showed is this certain ketone monoester that is a esterified radiohydroxybutyrate with 1,3-butanediol actually does increase exercise capacity in humans. And this is an interesting one because once it's deesterified, you get the 3-hydroxybutyrate burst at the beginning, but the 1,3-butanediol is converted to 3-hydroxybutyrate in the liver, so you get a sustained increase as well. And this is kind of data from her cell metabolism paper a few years ago. She showed the human subjects when they drink this ketone ester, they just put it in basically Gatorade. You can see an increase in blood radiohydroxybutyrate that gets in the 2 to 3 millimolar range. And most importantly, shown here in panels F and G, they found that this does seem to increase the distance run in this trial. So we wanted to see, well, do we see a similar thing in mice? And ask a specific question: does acute administration of ketone monoester increase exercise capacity, and is it dependent on cardiomyocyte BDH1? So we designed a kind of a complicated experiment that involved 3 different exercise capacity tests. Using the cardiac-specific BDH1 knockouts with their Cre-negative littermates. And what we did is wanted to see, okay, before we even delete the BDH1 gene, is there any difference in exercise capacity between these 2 genotypes? We can then delete the gene, giving tamoxifen and giving a washout period, and then see does deletion of the gene by itself have any effect on exercise capacity. And then finally, in the last one, we gave the ketone body monoester or water as a vehicle control to mice right before the exercise capacity test to see if the ketone monoester increases exercise capacity in the mice and if it's dependent on BDH1. And so for this, we measured work and distance. We had pre and post measurements of glucose, lactate, and ketone bodies. The experimenters were blinded to genotype and treatment. And this is the exercise capacity test that we use, in which it's a rather intense capacity test because you're increasing not only the speed, but also the inclines. The amount of work they're having to do over time is increased. And once the mouse fatigues, we take them off the treadmill. That's when we take all the measurements for the pre and post measurements of glucose, lactate, and ketone bodies. And then we can measure the actual work that they've done, the distance they've run as well. So this is kind of just showing one of the concerns with this type of capacity test. It can be kind of subjective when you pull the mice on the treadmill. Like, when do you know they're actually fatigued? We have very specific criteria for this, but it still takes a person to make those judgment calls. And so one of the things you can do to check to make sure they went to the same, they exercise at the same intensity, is look at circulating blood lactate at rest and at fatigue. And so this baseline is showing this ECT1 result. Post tamoxifen is this ECT2 result. And you can see in both genotypes, we got lactate up to about the same, they went up to around 5 millimolar at fatigue. So we think that mice will exercise the same degree during this test. So looking at ECT1 and ECT2, we did not see any difference or any effect of deletion of BDH1 at all on the relative distance. So there was no difference between ECT1 and ECT2. And if you compared the control glutamates to the BDH1 knockouts, there was really no change in the distance. This is very similar with no change in work either. It really got interesting when we started doing the ketone monoester experiment. So we ordered this ketone monoester from Delta G, which they deliver. They have a lot of scientists that use them to support the research. And so, but we wanted to make sure it was pure first. So we subjected it to mass spectrometry to make sure there wasn't just 1,000 peaks in here, and it wasn't, make sure it was pure. And so it was relatively pure. We see a few, there's some sodium addicts and such out to the right. And these are degradation products that are thought to just occur in the mass spec. So it's predominantly pure. So then we had to dose the mice and figure out what dose to give them. And we wanted, again, to get in the 3 to 5 millimolar range. So we diluted the ketone monoester in water, either making a 20% solution or 50% solution. And you gavage about 100 microliters per mouse. It ends up being around 12.2 millimoles per kilogram is the actual dose that gets us to this level. Which is the 20% monoester level. I'm sorry, the legend doesn't really match up, but this is the 50% ketone monoester that we finally arrived on to perform to use in our studies. And so these are actually the data that came out of that study, which we were quite surprised. We saw the ketone monoester in the control literates. We saw an increase in distance and work, but this was completely abolished when we have loss of BDH1 in the myocyte, suggesting that this increase in blood flow would appear to be important and appears to contribute to exercise capacity in these mice. And interestingly, again, we did lactate measurements to make sure they exercised the same levels, and they did. But what was very interesting is the 3-deoxybutyrate measurements, the circulating measurements in the control mice, the Cre-negative mice, we saw that the 3-deoxybutyrate went up to about 3 millimolar levels and at rest, and at fatigue it went down a little bit, but significantly, but not that much. But what you'll notice is in the cardiac specific BDH1 knockouts, there's about double the amount of 3-deoxybutyrate in circulation. So at face value, this appears to suggest that the heart is a major consumer of 3-deoxybutyrate, and it drops a little bit more with exercise. So this is kind of where we are with this project. We, within the grant, we're doing a lot of other studies looking at other metabolic pathways, how glycolysis, how fat oxidation influence myocardial blood flow as well. And we have a large aging angle in this grant as well. So our working model right now is that the myocyte metabolism of ketone bodies is required for ketone body-induced hyperemia. And what we think is that BDH1 is really critical for this, and that the myocyte consuming 3-deoxybutyrate is known to change multiple pathways, metabolic pathways. It decreases glucose uptake. It can have effects on lactate levels. And so we think there's some vasoactive metabolite that's extruded that leads to the increase in the perfusion. One of these could be hydrogen peroxide as well. It's known that ketone bodies increasing ADH in the mitochondrion quite a lot, and that could drive some mitochondrial derived ROS that may be a signaling mediator. What I didn't mention so much was the downstream effects, what is happening at the level of the vasculature. And this is something that Dr. Nestoriak has really kind of worked out to some extent, showing that if you give substrates that increase the NADH-NAD ratio in the smooth muscle, like lactate, this tends to activate these potassium channels and promoting outward potassium current, and promotes hyperpolarization, decreases intracellular calcium levels, leading to vasodilation. With respect to aging, we showed that aging promotes systolic and diastolic dysfunction, exercise intolerance. We see a large increase in BDH1 levels in the heart despite decreased levels, circulating levels of ketone bodies. Aged mice have decreased myocardial perfusion at baseline, and then ketone body administration appears to restore this perfusion to the levels seen in young mice. And with exercise, at least in young mice, oral delivery of the ketone body monoester increases exercise capacity. And we, in our project, we have studies to look at this in aged mice as well. So I wanted to keep it kind of short in case there were lots of questions, because this is a very interesting area for me. It's actually an area that I can see myself growing a lot as well, because I'm a biochemist by training. So I'm working with vascular physiologists now. So I'm by necessity having to be put in the physiology pool and learning it. And I really enjoyed that process so far. This work was done predominantly by an MD Ph.D. student, Kara Gallants, who's in the audience now. Yibing Nong and Matt were essential for doing a lot of this work. And as I mentioned, this was everybody that's bolded here had a major role in this project at some level. And I'd like to mention Steve Jones too. He's set up the core system and the CCS to be functioning really well. And this wouldn't be possible without that. So with that, I'll go ahead and close and take any questions you may have.
So this is what really wonderful and warms my heart, no pun intended, as a nuclear cardiologist who grew up doing myocardial metabolism. And we have a lot of tools that could actually help, and we are getting a PET center.
Oh, wonderful.
And so, being able to do, you had some carbon-11 acetate data, but that was outside. But having a radiochemist here doing radio labeling of the hydroxybutyrate would be really great.
Yes.
But I actually have a completely more on the cardiology side question for you. We gave up on dipyridamole. And we gave up on it because of the so-called dipyridamole non-responders that made adenosine way more accurate in terms of being a predictable increase in myocardial blood flow. So the real question is, couldn't you use adenosine? The issue, as you were presenting it, it could be that aging diminishes the amount of available adenosine dehydrogenase, okay, which is the enzyme that dipyridamole inhibits, so that the adenosine levels passively go up because it's not being metabolized. And so, actually it's adenosine deaminase, the enzyme. So we get around that just by giving adenosine intravenously, 100 micrograms per kilogram per minute, and you should get maximum coronary blood flow. Is it better than what we use clinically now, which is regadenoson, an A2A agonist?
Well, with respect to this phenomenon, the work versus blood flow, right?
Yes.
There's been a lot of studies looking at adenosine, and that does, if you block that, it does not disrupt this relationship between work and blood flow. Okay. Same thing they've done with nitric oxide, inhibiting all nitric oxide synthase. You don't disrupt that relationship. So they've kind of ruled that out for this particular relationship. But with the 3-hydroxybutyrate, typically in humans, you do see increase in work. It's increase in MDO2. We see trends in the mouse data toward increased work, but it's not significant. So it could be a useful tool, but I'm not sure. One of the things we want to do with these mice is test other agents. So just give them a norepinephrine challenge. Do they dilate normally there? Maybe to adenosine? I would think it would be a good experiment to do, because we would learn a lot from it.
So the other addition, and we'll open it up for other questions, but the other addition that should be FDA approved relatively soon, and you probably don't need it for the mice. FDA approval is fluorine 18 fluoropiridaz. It tracks with myocardial blood flow linearly. It's way better than anything we've ever had. Rubidium plateaus, the saprocine principle. It gets to a point where you get more than double. You can triple your myocardial blood flow, but you're not going to triple the uptake. Same is true with thallium, sestimidine, and tetraphosphate. But fluoropiridaz is really kind of like clinical microspheres. And what that allows you to do this kind of work before and after. You can actually do it with exercise, because the fluorine 18 half-life of 18 minutes means that it's going to be around long enough for you to be able to actually do your experiment, do the injection, and then image later. Right. So we've got a couple of
there's some options there. I think what Dinesh Kalra is doing now, he's got a trial or a study set up in which it's just going to take normal healthy subjects and give them either this ketone monoester or just the vehicle control, basically Gatorade, what have you, and look at blood flow before and after exercise. But he's doing this via echo contrast imaging, and he recently had Jonathan Lindner, who's the world's expert in that. So that's what he's set up for now using the Divinity dye. That's very similar to the dye we use in mice, basically the same thing. But these other modalities may be better in the long run. Have to see.
Yeah, and clinically, the pet centers really do give my absolute myocardial blood flow and coronary flow reserve.
Yeah.
At a level that seems to be more accurate than contrast bubble destruction and reperfusion. But
yeah,
it's
I hope we grow into that.
That'll always be a nuclear versus echo. Right. Any other questions?
Brad,
Yes.
very good presentation. I have a question like the BDH knockout mice, there was no change in the exercise capacity. or the
Not
exercise-
without the ketone monoester. Yeah.
So under the baseline, so how does that kind of like jab individual as to these ketone bodies to if they are?
Yeah. So I mean, I think it's during exercise, ketone bodies don't go up that much. So this would be more of an intervention to improve exercise capacity on the, if they improve blood flow, we think maybe it may improve exercise capacity. But yeah, they do not have any change in exercise capacity just by deleting that enzyme. We see in mice, the ketone bodies go up about 1 hour after exercise, mainly in females. But we don't know the nature of that response, why that happens. Females in general have a greater ability to make ketone bodies. And so like during pregnancy, which is what the work Helen Collins has been doing, even if you just fast female mice, they have higher ketone body levels compared to males. And during pregnancy, you get higher ketone body levels that occur in the late trimesters period. So we think maybe that in the context of pregnancy, it may be important, but for what I've showed today, I think it's more of like an intervention to try to improve exercise capacity. And we're thinking it may be an intervention that we've used in aging population. This is making a stretch right now from the mouse data, but maybe it'll help them exercise for people that can't.
And these ketone bodies are reduced in the aged individuals. I think you showed the data.
Yeah, well, that's one of the things that I was very hesitant to say that, because there are a couple cohorts that we didn't see much of a change. It's always trending lower. But then that one that I showed, that one experiment with numerous mice, it was lower. But I think it depends on the stage of aging, what have you. So I'm not making the claim that, a 60-year-old man would have lower ketone bodies if you fasted him or something. But it appears at least that the heart expresses more of the BDH1 enzyme, kind of priming him to use them if they were available. Yes.
Have you done anything to identify the metabolites of hydroxybutyrate?
That's the big black box. That's the one that we do have some experiments in the grant to do that. So we're looking at, we think it's hydrogen peroxide is one of those that, if you look at coronary flow and plot it against hydrogen peroxide, it's completely linear, just like the relationship there. So we think that ketone bodies are probably increasing hydrogen peroxide. It could diminish lactate uptake, which lactate's a known vasodilator as well. But I've always had the question, well, if that's the case, with exercise, you get increases in lactate in circulation anyway. Why would you need it from the myocyte? So I think it maybe take a couple metabolites to come together to make this happen. And actually lactate will, if you have hydrogen peroxide there, with lactate, you get a much bigger vasodilatory response just in the isolated vessel prep. If you have pyruvate there instead of lactate, there's no vasodilation. So there's a redox mechanism involved. We know that. And we're trying to figure out what this metabolite or metabolite cocktail may be that's leading to the coronary vasodilation. It's a hard problem. It's a hard problem, actually. And that's what one of the reviewers asked us, that what are the metabolites? But we're not there yet.
So you said that these ketone bodies come from metabolism in the liver.
Mhm.
Is there anything with the microbiome?
Well, ketone bodies, they can be produced also by the kidney to some amount, but also microbiome can make some ketone bodies as well. So those are, two other possible sources. The majority of it's made by the liver, though.
Well, this was really spectacular, and I guess we give people back 18 minutes
Yes.
of it, and when we get to do Cardinal Research Minute.
Yeah.
So thank you very much.